Yeah, hello, my name is Juergen Goetz. With my team, I'm based at the Clem Jones Center for Aging Dementia Research at the Queensland Brain Institute of the University of Queensland in Brisbane, Australia. And here I would like to talk about improving the safety profile of focused ultrasound for blood-brain barrier opening by applying a combinatorial strategy that involves the Claudin-5-specific binder. In my lab, we work with ultrasound, basically applying three modalities. We use ultrasound on its own, which we call scanning ultrasound only. We use ultrasound with microbubbles to achieve blood-brain barrier opening, and we combine this modality with monoclonal antibodies. And we basically apply this technology to wild-type mice, but also to Alzheimer's mouse models. And applying these technologies or these modalities, we basically deal with two fundamental ultrasound principles. One is the application of a radiation force. The second is the application of cavitation. We believe that for the conditions we are applying, that heat can be neglected. Now, when we look at low-intensity ultrasound, we find that ultrasound, as reviewed by us 2 years ago, obviously has an effect on by opening the tight junctions, allowing therapeutic molecules to enter the brain. But at the same time, there are also more vesicles forming in the endothelial cells. So in other words, there's on the one hand a disruption of tight junctions, as shown on the right, but also an upregulation of transcytosis. And together, this permits the transport of cargoes, as shown in green, for therapeutic molecule across the blood-brain barrier. Now, the question arises when one takes this into an in vitro situation, which two-dimensional blood-brain barrier systems are available to study the effect of focused ultrasound. And as reviewed just recently, there are human models available, there are canine models available, and murine models available. And on the right, I've listed the key features these cell types need to have. On the one hand, they need to express tight junction proteins, such as Claudin or Claudin, GEM1, ZO1. These cells need to have a cobblestone morphology, and we need to have a high TR value, high trans-electrical interfield resistance, which can be assessed in these transwell inserts. And on the left, you see the different values depending on which system one uses. For example, when one uses induced brain endothelial cells on the left, these cells have a very high value of 2000 to 4000 ohm square centimeters. On the other hand, when we look at MDCK cells, the values are much lower, but we have in the meantime generated a cell line, as I will discuss, that has a higher TR value. So there's a compromise between having very high TR values and availability. Now, the idea which we had was to use a Claudin-5 binder as a wedge, allowing us to use lower acoustic pressures to achieve a comparable barrier opening, with a possibility that this could be eventually potentially translated. We published this work in Theranostics and also featured the cover image. The Claudin-5 binder enhances focused ultrasound-mediated opening in an in vitro blood-brain barrier model. So basically, we have these MDCK cells. As I said, they have canine origin. They have a TR of 200. They express Claudins, but not Claudin-5. And therefore, we introduced a vector where Claudin-5 is expressed, a human Claudin-5 tagged with an HFP, such that Claudin can be visualized. This allowed us, as shown on the left bottom left, to increase the TR value threefold. You can also appreciate that Claudin-5 is now located to tight junctions. You see also areas where cells are not in cell-cell contact, and hence there's no localization of Claudin-5. And obviously Claudin-5, not only do these cells show a cobblestone morphology, but Claudin-5 also co-localizes with occludin, and we showed this also for ZO1. Obviously, as I've said before, the TR values are much lower as for induced brain endothelial cells, but obviously MDCK cells are also a much easier cell line to work with. It's more economical and they are more available. When we looked into the literature, we found that there are a few molecules which have been described, which are shown to weaken the blood-brain barrier. There's a work from Dittmar and colleagues published in 2010. They designed peptides targeting Claudin-5, including MC5C2. This peptide binds the extracellular loop domain 1 of Claudin-5. And an earlier paper from Potts and colleagues published in Journal of Cell Biology in 1999, where we used the mutated non-toxic domain of an enterotoxin, which we've used the GST. And this truncated protein binds to the second loop domain of Claudin-5. And then we tested these two compounds in our newly generated MTCK cell line and found that in a dose-dependent manner, it achieved blood-brain barrier opening as indicated by a drop in TEER, but also by an uptake of cargos, data I'm not showing. The same obviously can be done with focused ultrasound. So we take the same cell system, we use focused ultrasound, we use particular pressures, you see 0.3 and 0.4 megapascal, and we see that after ultrasound, the TEER value drops. And we also find that with time, the barrier closes again. What we then did is we pre-incubated these MTCK cells that express Claudin-5 in a tagged form, and pre-incubated these cells either for 2 hours or for 12 hours with this Claudin-5 binder, and then applied ultrasound and measured baseline TEER and post-treatment TEER, and also looked at FT40 plate readout to get cargo passage, cargo leakage. And we found that, as shown here on the left, with our data which we published, is that when you look at the different pressures which we applied, that at 0.1 and 0.2 megapascal, the TEER was significantly lower when the cells were treated with combined focused ultrasound and the Claudin-5 binder. And these changes which we find for the TEER values were also reflected by changes in permeability, in that the leakage of FT40 was increased. This is something we are currently testing in vivo. And the take-home messages, as summarized in Advanced Drug Delivery Reviews just a few weeks ago, our data suggests that pre-incubation with clinically suitable binders to tight junction proteins, possibly also to adherence junction proteins, may be a general strategy to facilitate safer and more effective ultrasound-mediated blood-brain barrier opening in cellular and animal systems, and potentially also for the treatment of human diseases of the brain. With this slide, I would like to thank those who have contributed. I've listed my collaborators, but also research support we have received, and from my team, and particularly Juergen, who has been driving this project. Thank you.